do a quick acknowledgement of anyone who is watching this recording or watching this as it's being live streamed in other places. It is my absolute pleasure to introduce our first keynote speaker, Dr. Cassandra Extravour. Cassandra is a full professor in the Department of Organismic and Evolutionary Biology at Harvard University. She has a PhD in molecular biology from the Autonomous University of Madrid. And during the course of her career, she has received and been nominated for many awards, including the Joseph R. Levinson Memorial Teaching Prize and the Ellison Medical Foundation New Scholar Aging Award. Today, she'll be talking to us about her research, which is focused on understanding early embryonic development and the origins of these genes that control this development and how they change, their functions have changed over time, with a particular focus on the reproductive system. And then she will also be speaking to her experiences through her career as a woman in STEM. And so with all that said, let's all give a warm welcome to Cassandra.
Hi, everybody. If you'll just bear with me 1 second, as I was telling Denisa before we started, I've been having some real technical issues, and I know that sounds lame, and it's 2021, and I shouldn't be having any technical issues, but that's the life right now. So I just need 30 seconds to see if it's going to be possible for me to work this out so you can see my slides. And if not, I'm just going to talk for a little while, because the most important thing is that you all be able to ask me any questions, that you might have about the work or about science, or anything that I can help with. So that's really the most critical thing that I want to do. But I don't like to give up. And so I'm trying one last ditch attempt to see if PowerPoint is going to cooperate with me here. Let's see. Just about. Okay, let me try this out now. Let me try and do a screen sharing. Oh boy. All right, what are you all seeing right now?
We can see your slides.
Okay, it's just like it's blank, but it has a title at the top?
Yes.
Okay, great. So
looks great.
Thank you. All right. I want to thank Denisa for the intro, and thank Linnell for getting this project off the ground so many years ago, and thank all of you for being here. As I said, I'm going to try to keep my remarks pretty brief, because I want you all to be able to ask me any questions, because you know what you want to know about me or from me. But I'll give you a little bit of context so you know a little bit about me and where I'm coming from. So I'm a professor, as she said, and I'm a scientist. Is what people ask me what I do. I say I'm a scientist. That's really the thing I'm most interested in. At the end of the day, I end up spending my time on a lot of things that aren't science, sort of to create an infrastructure where we can get science done. And so the science that we do in my lab this is how I see it, as you're going to see on this first slide. I formally work on developmental genetics, which means what are the genes that tell cells to do things correctly or incorrectly if they make a mistake during the process of development. And by development, I mean going from a single cell to a multicellular organism. It could be a dog, could be a tree, could be a mushroom, doesn't matter. In my lab, we end up working with animals, and the specific animals we work on, as you'll see, tend to be insects. It's for convenience reasons. We can manipulate them easy. It doesn't cost a ton of money compared to other organisms. Because really what I'm fundamentally interested in is this relationship between genetic information and ultimate cellular outcomes. The reason that I think it's very interesting to study this in a multicellular context is because stable multicellularity, stable obligate multicellularity, and by that I mean the condition of cellular life that requires that many cells be together and that they stay together in order for the species to reproduce. So we, for example, we humans are obligate multicellular organisms. We don't function as organisms unless all our cells are physically stuck together, and our species can't reproduce. We can't reproduce as individuals, and so the species can't reproduce unless we are a whole individual, right? Not all life is like that. In fact, the majority of cellular life on Earth is unicellular. Just one cell at a time, one bacterial cell, one fungal cell. Their reproductive cycle is just a cell cycle, mitosis. They divide, that's it, a new generation. And yet, This condition that is stable multicellularity evolved many, many times. By many, I mean like at least 10 or at least 20, depending on how you analyze it. And what's interesting to me is that every time that stable multicellularity evolved, you also evolved another aspect, not just lots of cells stuck together, but different cells now have to do different jobs. Every cell cannot be identical in function. Every cell has a unique function to carry out for the whole organism to be successful. The function I find most interesting is the function of the cells whose DNA will actually make it into the next generation, and those are the germ cells or the germ line or the propagules or whatever you want to call it. These are the cells in our species. These are cells that make eggs and sperm. Without these cells, evolution would come to a halt of our species because no other cell in our body is capable of making another generation of humans, only those cells. Interestingly, every time stable multicellularity evolved in eukaryotes, the germline also evolved some kind of sort of narrow, bottlenecking, small number of cells, type of cell lineage or cell type in the organism. And this is a repeated feature of the evolution of multicellularity. And then with the evolution of reproductive division of labor, some cells will make another generation, the other cells will just be around for one generation, and then that's it. With that also evolved then the evolution of different types of somatic cells. The somatic cells are all the cells that aren't the germ cells. So you have your skin cells, you have your liver cells, you have your hair cells. These are all different types of somatic cells. So the evolution of multiple cell types is also a stable feature that involves first the appearance of a germline, and then we get diversification of the soma. So for me, the developmental cycle or the developmental process that we try and understand is really a kind of a manifestation of the whole evolutionary process when it comes to evolving this feature of cellular life, which is multicellularity, which has happened so many times on Earth. And so that's how I think about development, and that's how I think about evolution. And when we study this in my lab, our approach is to try to understand how this could have occurred, right? We're trying to understand an evolutionary process that part of it is past, part of it is ongoing, and we'd love to understand or predict what's going to happen in the future. And so we know that to look at this big problem, we cannot only take one approach, we cannot only study one organism, we can't forget or ignore how organisms are related to each other. In every piece of information we learn about how any given organism does anything, we have to contextualize it in the context of the rest of evolution. And so that's why my lab ends up in practice working on a lot of different organisms, a lot of different types of cells. We use a lot of different tools and techniques because we're trying to study this problem in a big way. As I said, the study systems that we end up working on in practice in the lab, a lot of the time are arthropods. So arthropods are now and always have been the most abundant, dominant type of animal on the earth. And they are insects and spiders and myriapods and stuff like that. They also happen to contain the most highly manipulable genetic model organism that is an animal, and that's a fruit fly, Drosophila melanogaster. So we can perform really like unparalleled in any animal system genetic manipulation of one of these animals, and then for the others we try to manipulate them as best we can a little bit as well. So I'm not going to do a deep dive into any specific science story, but I'll just tell you that I think a general theme that emerges from the sort of work that we do ends up being scrutinizing, maybe in a little greater detail, maybe in a little greater historical context, maybe with more different types of tools, maybe in more different types of organisms, big claims that often appear in the literature about sort of the way things work, quote unquote. And so one of them, for example, is these special cells, these germ cells that I talked about, whose genetic material has to make the next generation. How does a developing embryo make sure that those cells get established in the first place? How does the germline become specified? For a long time, I mean, hundreds of years, there was a sort of dominant thinking that there was one specific type of mechanism that made sure that a germline appeared, and it had to do with a special type of cytoplasm that was inherited by cells called germplasm. But this conclusion that this was sort of the way, quote unquote, that animals did it was based on the study of a really small number of animals. And to sum up a long period of time in my research career, if you look at a lot of animals and you really look at representatives of all the animals, this germplasm method is not the most dominant way. It's not the most common way. And we don't think it was the ancestral way that animals made sure that they had a germline either. So with these different contributions, we've been able to contribute to changing the textbooks that used to say, all animals make their germ cells in the following way. And now they say, looks like most animals make their germ cells in this way, and that's probably the ancestral way that animals did it. And then in some animals, a different way evolved. And that happens to be the way that we understood the most in the 20th century. Another recent line of research that we've gotten into concerns the evolution of shape, the evolution of size, right? Organisms have a certain characteristic size. It's not fixed. It's kind of, there's a median and there's some kind of range. And they also have a median shape, right? Again, it's not fixed, but shapes and sizes are characteristics of many types of organisms, big frogs, small frogs, whatever. So how do you determine shape and size? How do cells know how many of them they should make, what shape they should arrange themselves into? People have been fascinated with this question for a long time. People love the idea of universal laws that explain everything, generalizable principles that explain everything. And there was a thinking for a long time that when it comes to sizes and shapes of things made out of cells, so cellular structures, that there were universal laws that governed the sizes and shapes of things that cells could make. These laws predicted that certain shapes could only exist in certain size ranges, or that when certain sizes of things evolved, their shapes would have to change to accommodate those things. Again, long story short, we don't think this is right. We took one case study. You can't study everything. We took one case study, which is eggs. The eggs of insects. Eggs of all animals are single cells. They come in massive ranges of sizes. Human eggs are, 50, 60 microns. We study eggs of beetles that are like the size of a blueberry, like a centimeter. So it's orders of magnitudes larger than other cells, even within just the insects alone, which is all we considered in these recent studies. 8 orders of magnitude difference, super, super tiny cells, super massive cells. They're all the same type of cell. They're all egg cells. They also, as you can see in this little picture, make very different shapes. Some are perfectly round, some are super long and thin, some are curved like bananas, some are flat like a little disc. And we found that essentially, if you consider a large enough sort of chunk of life, and you keep in mind critically how these organisms are related to each other evolutionarily, you find that there are no laws. There are no universal rules that predict the sizes or shapes of certain things. And then finally, another thing that we've sort of looked into, and the more we looked into it, we thought, wait a minute, we have to rethink how we used to think about how new genes get made. We've been studying this gene. The name is not important. It's an interesting gene that it's important in the fruit fly and in some other insects to make germ cells that I was starting with at the beginning, right? So for a long time, this gene, which was discovered in the fruit fly to be critical for making germ cells, you absolutely needed it to make your germ cells. And in fact, just having it, just expressing it could make you become a germ cell if you didn't already express that gene. This gene was for 30 years thought to be a germline gene, meaning its role is in the germline, it's required for the germline. Germline can't do without it, and it doesn't have any other job, just this germline job. Again, long story short, we found that that's not true. This gene does have different jobs that are not only in the germline, not just in the fruit fly, but also in other insects. From an evolutionary point of view, this gene was thought to be something like 300 million years old. We recently, again, looking deeper, showed it's got to be at least 450, maybe 500 million years old, so a lot older than we thought. And it is not used to make germ cells in many, many insects. It's used for something completely different. And probably ancestrally, when the gene was first born, when it first showed up in the first genome of this ancestral insect, this is our hypothesis, didn't have anything to do with the germline. It was playing a totally different role. And then through a series of changes over evolutionary time, it gained a role in the germline of some insects that happens to be the role that we humans discovered first, but that wasn't the ancestral role. And then one of the more surprising things that we found out about this gene, again, by looking very carefully at the sequences, is that the most likely origin, the most likely explanation for where this gene came from, because the gene doesn't look like any gene from any other animal. We think that this gene came from a fusion of sequences from a bacteria and from an insect or from some kind of animal. These two pieces of DNA from two different domains of life fused together and created a gene with a brand new structure, which is why we think nobody could identify who Oscar's relatives were for decades, because it doesn't have any relatives from animals or from bacteria, because it's a fusion of these two types of sequences. Okay, so this is just like a big sort of overview of the sort of flavor of perspective that I take on science and some of the outcomes of it. So now in, a couple of minutes, let me just give you a sense for sort of where I'm from and what my perspective is in a broader sense. So I was born in Canada. My parents are immigrants to Canada, both came through different routes. My father is from Trinidad and Tobago in the Caribbean. He immigrated to Canada as a young man, intending just to study briefly to finish up a correspondence course he was doing with a community college. And there was a requirement to do the last course of the certificate in Canada, but then he met my mother in Toronto when he moved there and decided to stay there. And my mother was in Toronto because her family had emigrated from Switzerland, where she was born during the Second World War. After the war was over, my Hungarian grandfather and my Swiss grandmother decided they wanted to make a new life and get out of Europe. They ended up in Canada, brought my mom with them. So my mom spent, her later childhood years and adult life in Toronto. My parents met and raised 4 kids. I'm the oldest of 4 kids. I have a brother and 2 sisters. My brother became a scientist and now he works in science policy. My older of my 2 sisters is a vegan chef and she moved to Trinidad. She always hated Canada. So she moved to Trinidad where our family is from and she's living there. And then my other sister stayed in Toronto and wasn't sure what she was going to do. And she settled up doing a combination of accounting and bookkeeping and singing. So we all do different things. And like a lot of immigrant families, my parents' goal for me was get more education than we did. They had high school education and my father had that community college certificate. They hadn't been able to go to university or complete university degrees. They really wanted us to do that. So they encouraged us to do that. My brother and I did that. My sisters, the university path was not for them. They've made their very successful lives on their own separately. So I did my bachelor's degree at the University of Toronto. I'm from Toronto, so I didn't have to move. I could walk to the University of Toronto because I lived downtown. And I had jobs to pay for school. Since the age of 12, I was working full-time in the summer and part-time in the school year. Went to University of Toronto. And then for the first time when I was there, heard or sort of learned that going to school for money was possible and that it was called doing a PhD. So I can answer any questions that you have about this later, but I discovered that I was interested in science at a very late age. I wasn't interested in science as a child. I became interested in science when I was almost out of high school. In undergrad, I almost left biology and went into math because biology seemed pretty boring to me at first. But the important thing I learned, I would guess, during undergrad is that I could go to grad school. I had heard of PhD, but I didn't know what that meant or what it was for or how you did it or what you did with it. But then I learned that was basically like advanced school, and I was a good student and I enjoyed school and I enjoyed learning. And so my understanding of a PhD is I'm going to get paid for 4 to 6 years to learn something that I think is interesting. And I'm going to get paid enough to pay my bills. I didn't have dependents or children, so I needed only a small amount for my own rent and so on. And my understanding was I won't have to waitress or be a secretary or work at McDonald's anymore, which is what I had been doing, because the grad school stipend will be enough. So I just have to get into a grad school. And with some help from professors of mine who gave me suggestions of people I should contact, I ended up in graduate school studying science and doing a second thing that I had wanted to do, which was living in a different country and living in a different language. I moved to Spain for my graduate work. And I think I'm going to stop talking there because this slide sort of summarizes that. But my graduate work in Spain was in developmental genetics. And at the beginning of grad school, I just wanted to know how genes worked. And by the end of grad school, I wanted to know how genes evolved and how their functions evolved. And so that's why I took up postdoctoral studies, which I hadn't heard of before I was a PhD student. And then I was like, oh, I can keep getting paid to learn more science. It's called a postdoc. Great. And I have discovered I love the experience of moving to a different place, moving to a different country. So I decided I was going to keep that going. So I moved to Greece briefly, and then I moved to Spain, to England, and did postdocs there. And then I wasn't really sure if I wanted to have my own lab, but I thought maybe I would like it. And without thinking too much about it, honestly, I applied for the job that it turns out is still the job that I have now, which is a faculty member at Harvard. So I'm going to stop there because I'd love to take your questions and hear what it is that I can how I can be most helpful to you guys today. So I'm going to stop sharing.
Okay, thank you so much for that incredible presentation. I wanted to say a quick note that for people who are live streaming this, the live stream will end now so that we can accept questions from our participants and people in the webinar. And so you can join us again tomorrow with our next live stream keynote, which is Malika.